Welcome to this lecture on synaptic plasticity. Have you ever wondered how your brain actually stores a new memory or learns a new physical skill? Today, we are going to explore the microscopic changes that happen inside your head every time you experience something new. This is one of the most fundamental and exciting topics in all of neuroscience. To begin understanding how our brains change, let us imagine you are walking through a dense, overgrown forest for the very first time. You have to push heavy branches out of the way, and your progress is incredibly slow. However, if you walk that exact same path every single day, the trail eventually becomes wide, clear, and easy to travel. On the other hand, if you suddenly stop using that path for a year, the weeds and bushes will grow back, making it difficult to pass through once again. In the human brain, the connections between our brain cells behave exactly like these forest paths. The more a specific connection is used, the stronger and faster it becomes. Now that we have a mental picture of how pathways strengthen, let us look at the actual biology. In the field of neuroscience, synaptic plasticity is formally defined as the biological ability of synapses to strengthen or weaken over time in response to their activity levels. But what exactly is a synapse? It is the tiny communication gap between two neurons. The first neuron, known as the presynaptic neuron, releases chemical messengers across this gap. These chemicals then travel to and activate the second neuron, which we call the postsynaptic neuron. This dynamic, ever-changing nature of neural connections is what allows our nervous system to physically rewire itself based on our experiences. Building on our understanding of the synapse, let us explore how these connections actually get stronger. When the presynaptic and postsynaptic neurons fire electrical signals at the same time repeatedly, the synapse undergoes physical and chemical changes to make future communication more efficient. This strengthening process is known as long-term potentiation. Physically, this can involve the presynaptic neuron releasing a larger amount of neurotransmitters into the gap. Alternatively, the postsynaptic neuron might build more receptors on its surface to catch those chemical messengers more effectively. This is the biological equivalent of clearing the brush on our forest path, making the signal travel much faster and easier the next time. While strengthening connections is vital, the opposite process is equally important for a healthy brain. If the two connected neurons rarely fire together, the connection is actively weakened through a process called long-term depression. Just like the forest path becoming overgrown with weeds when abandoned, a neglected synapse loses its efficiency. The postsynaptic neuron might remove receptors, making it less sensitive to incoming signals. This weakening is not a flaw. It is a crucial feature. It allows us to forget useless information, break bad habits, and prevent our neural networks from becoming dangerously overactive. Without long-term depression, our brains would be overwhelmed with unnecessary connections. Understanding these mechanisms of strengthening and weakening leads us to why this topic is so revolutionary. Synaptic plasticity is arguably one of the most important discoveries in modern neuroscience because it provides the physical explanation for how learning and memory actually work. Every time you learn a new language, master a chord on the guitar, or memorize a friend's phone number, your brain is actively using synaptic plasticity to build and strengthen specific neural circuits. If our brains lacked this incredible ability, we would be completely trapped in the present moment, unable to form any new memories or adapt to changes in our environment. Beyond everyday learning, This concept brings us to its critical role in medicine. Understanding synaptic plasticity is absolutely crucial for treating severe neurological conditions. For instance, diseases like Alzheimer's involve the pathological loss of these vital synaptic connections, leading to cognitive decline. By deeply studying how synapses strengthen and weaken, scientists and doctors are developing cutting-edge therapies. These treatments aim to help stroke victims physically rewire their brains to relearn lost motor skills and to prevent debilitating memory loss in the elderly population. It is a field of study that holds the key to curing many brain disorders. To understand this biological process mathematically, we use a simplified model known as Hebb's rule. Named after Donald Hebb, who famously said that neurons that fire together wire together, this formula captures the essence of learning. The symbol delta W represents the change in the synaptic weight, which is the physical strength of the connection. The symbol eta represents the learning rate, a small constant determining how quickly the synapse adapts. The variable x is the firing rate of the presynaptic neuron, and y is the firing rate of the postsynaptic neuron. If both x and y are highly active simultaneously, their product is large, causing a massive increase in synaptic weight. If either is inactive, the product is zero, meaning no strengthening occurs. Now that we have our formula, let us see this with a concrete example. Imagine a student practicing a new piano scale. We have a presynaptic neuron representing the visual input of the sheet music, and a postsynaptic neuron representing the motor command to move a specific finger. We're given a learning rate, eta, of 0.1. During a successful practice, the presynaptic neuron fires at 50 spikes per second, and the postsynaptic neuron fires simultaneously at 40 spikes per second. We substitute these values into Hebb's rule. We multiply 50 and 40 to find the combined simultaneous activity, and then scale it by the learning rate of 0.1. The final change in synaptic weight is positive 200 units. This means the connection has grown significantly stronger, forming the biological basis of muscle memory. This basic formula we just used leads us to an important question. What stops connections from growing infinitely strong? Pure Hebbian learning has a flaw because weights would grow forever if they only ever increased. To fix this, neuroscientists use a modified rule where the postsynaptic activity must exceed a certain threshold to cause strengthening. In this modified formula, we introduce the symbol theta, which represents this biological threshold. We subtract theta from the postsynaptic firing rate, y. If the postsynaptic neuron fires faster than the threshold, the term is positive and the synapse strengthens. But if the postsynaptic activity is lower than the threshold, the term becomes negative, which mathematically models the weakening of the synapse, or long-term depression. Before we move on, let us apply this modified rule to see how a synapse weakens. Suppose a synapse has a learning rate, eta, of 0.05 and a threshold, theta, of 30 spikes per second. The presynaptic neuron fires at 40 spikes per second, but the postsynaptic neuron only fires at 10 spikes per second. We substitute these values into our modified equation. The critical part is evaluating the expression inside the parentheses first, following the standard order of operations. 10 minus 30 gives us negative 20. This indicates a failure to reach the threshold. We then multiply 0.05 by 40 and then by negative 20. The final change in synaptic weight is negative 40 units. Because this value is negative, it physically means the synaptic connection has been weakened. Now that we have calculated these changes mathematically, Let us look at a visual representation of what is actually happening microscopically. On the screen, you see a diagram comparing a synapse before and after learning. On the left, in the weak state before learning, the top presynaptic neuron releases only a few red neurotransmitters, and the bottom postsynaptic neuron has very few blue receptors to catch them. Following the arrow labeled synaptic plasticity, we see the strong state on the right after long-term potentiation has occurred. Notice the dramatic physical changes. The presynaptic neuron is now packed with red neurotransmitters, releasing them in large quantities. Simultaneously, the postsynaptic neuron has built many more blue receptors along its surface. This structural upgrade is what makes the neural pathway faster and more efficient. To wrap up our lecture, let us review the most important ideas we have covered today. First, synaptic plasticity is the remarkable ability of the brain to continuously mold and adjust the strength of its connections based on your daily experiences. We learned that through long-term potentiation, active neural circuits are upgraded and strengthened, while long-term depression ensures that neglected or uncoordinated connections are pruned away. We also saw how Hebb's rule mathematically models this process, proving that neurons that fire together wire together. This microscopic dance of chemical and structural changes ensures our brains remain adaptable. You now understand the ultimate biological foundation for every memory you cherish and every skill you possess.